This Week in Microbiology is brought to you by microbe.tv and is supported by the American Society for Microbiology. To learn more about microbes, visit microbe.tv and asm.org.
This is TWiM, This Week in Microbiology. I'm Michael Schmidt from the Medical University of South Carolina, and joining us today on our podcast where we explore the unseen life on Earth from Ann Arbor, Michigan, is Michelle Swanson. Welcome,
Hello.
Michelle.
Suddenly it's September.
It is
good to be
indeed.
with you.
And also joining us today is our TWiM adjacent colleague and host of Matters Microbial that drops every Thursday, the ASM Karski Award-winning professor of microbiology from the University of Puget Sound, Dr. Mark Martin. Welcome, Mark, and thank you.
It's always good to be here. I so enjoy these discussions.
Vincent and Petra are away at separate meetings this week, but we are still going to offer you some exciting science, which takes me to our first paper, a snippet entitled Vibrio pectinsidia strain FHCF3 is a causative agent of C-star wasting disease. And this paper is by 15 authors. And I thought it would be fun as we begin our new academic year for our snippet this week to select a paper that effectively takes us all back to our microbiology roots. And the paper comes to us from Nature, Ecology, and Evolution. And as I said, is authored by 15 individuals where they solve a huge mystery that's been plaguing our Pacific Northwest coast of North America for over a decade. Today, we're going to be a witness to a classic case of microbial detective work, fulfilling Koch's postulates and finally pinning down a culprit for this devastating C-star wasting disease. So what's this problem? What is wasting disease? So back in 2013, a massive epidemic called C-star wasting disease, or SSWD, started wiping out sea stars all the way from Mexico to Alaska. And sea stars are what you might expect. They look straight out of SpongeBob SquarePants. It's the starfish that we all grew up with, or your kids grew up with, or your grandchildren grew up with. And we're talking about the largest documented marine epidemic of a non-commercial species. It affected over 20 different species of these sea stars, but the hardest hit was the sunflower sea star that you can see in an image associated with the companion piece by Kevin Lafferty in the Nature's News and Views section of Nature, Ecology, and Evolution. And the sea star's genus and species name is Pycnopodia hylanthioides.
And Michael, if I could jump in, kind of really illustrating the One Health concept, the loss of this particular species of starfish had dramatic impact on the whole food chain in the environment, which I imagine you're going to talk about.
Precisely, Michelle. These stars are, or were, major predators of the ecosystem. So they were extremely important. Once you eliminate a predator, the ecology of that environment really falls off the tracks. And their disappearance, billions of them, where they died, triggered a devastating trophic cascade. Without the sunflower stars to eat them, sea urchin populations exploded, and they literally consumed entire kelp forests of the Pacific Northwest coast. And the sunflower sea star is now considered functionally extinct in much of its southern range and is listed as a critically endangered species. For years, scientists have been trying to figure out what was causing this massive die-off, with early theories pointing to a densovirus. But since this is TWiM, we're going to invoke bacteria. And it wasn't a virus. I'm just going to cut to the chase. And that's because the evidence never quite held up. So the investigation is really classic microbiology. And this is where this new study comes in. The researchers, led by Melaine Prentice and Alyssa Lois Gemin, took a very methodical approach to find the causative agent. First, just like Coke, they showed the disease was transmissible. They took tissue extracts, cholamic fluid, that's the fluid in the sea star's body cavity, and even just the water from the tank with sick sea stars, and they used that fluid to expose to healthy ones. Sure enough, the healthy stars got sick. They developed the classic signs of this wasting disease. Their arms of the starfish would twist and curl, and then they would literally just detach, a process called arm autonomy. And then within about 2 weeks, the poor starfish would die. The control groups, which were exposed to heat-treated versions of the same materials, either tank water or this cholamic fluid, remained perfectly healthy. This
and
told
their
them,
figure 2 illustrates this beautifully.
Oh, absolutely.
Just beautiful color photographs. You can see the arms starting to curl, and then the whole thing is necrotic.
And I kept channeling SpongeBob SquarePants, thinking what SpongeBob the Sponge would be saying at this time. And next, as you might imagine, they did deep sequencing, something that Koch didn't have available to him back in the 1860s. And they did it on the cholamic fluid from both sick and healthy stars. They saw a clear signal that popped out from the disease samples, a massive number of reads assigned to a bacterium identified as Vibrio pectinsidia. In some disease stars, this one bacterium made up between 23 and as much as 99% of all the bacterial reads that they found in this cholamic fluid. While they could find tiny trace of it in some healthy appearing stars, the abundance was dramatically higher in sick ones, pointing to it as a strong candidate that will then go on to fulfill Koch's postulates. And this is the really cool part. They just didn't stop at finding an association. They went the full 9 yards. They went on to fulfill Koch's postulates. First, they found the microbe in the disease but not healthy individuals. Then they did this with their sequencing data, showing a strong correlation between high levels of Vibrio pectinsidia and the sea star wasting disease. Then they isolated the microbe, and they were fortunate enough to grow it in pure culture. Now, any of you who have worked with environmental isolates like the esteemed Dr. Martin know that's no mean feat. That's pretty hard to do, to work with organisms from the ocean, because you can't go to the shelf and pull off LB for sea star wasting disease. So they did a really fantastic job, and the researchers successfully isolated and cultured a specific strain, which they named FHCF-3. And this came from the cholamic fluid of the diseased sunflower sea star. The cultured microbes should then cause disease when introduced into a healthy organism. Folks, this is the smoking gun. They injected healthy quarantined sea stars with the pure culture of Vibrio pectinsidia, strain FHCF3. The injective stars developed the exact same signs of sea star wasting disease, arm twisting, autonomy, and ultimately death, while the control stars injected with a heat-killed version of the culture remained perfectly normal. They even showed a dose-dependent response, where a higher dose of the bacterium caused the disease to progress faster. And that makes perfect sense from an intuitive sense. Then they re-isolated the microbe from the experimentally infected host, and finally, they were able to re-isolate Vibrio pectinsidia from the sea stars that they had experimentally infected, confirming its identity with sequencing. So they nailed every one of Koch's parameters. They demonstrated that the Vibrio pectinsidia strain FHCF3 is indeed the causative agent of the sea star wasting disease.
And another neat control they did was ask whether, if they put the solution through a filter, a 0.22 micron filter, then the fluid was then no longer would cause disease. So they
No
were able to
longer
deduce
infectious.
then that it was not like a molecular toxin, but rather something large, a cell, a bacterial cell, it turns out.
Also not a virus.
Not a
Right,
virus,
right.
because viruses are filterable agents,
Yeah.
as we learned. And this discovery is a huge step forward for marine biologists. For a decade, we haven't known what was driving this ecological collapse. Now that we have a culprit, researchers can start developing strategies to try to mitigate the disease. They can screen for the pathogen's presence in the environment, study how factors like warming temperatures might influence outbreaks, something often linked to pathogenic Vibrio species, and the One Health hypothesis that has been developing over these past 3 decades really implicates climate change in a lot of the outbreaks that the One Health investigators have been studying. They're also looking to work on recovery efforts for the sunflower sea star because of its importance in keeping kelp forests healthy that depend on it. This is, really a fantastic piece of both modern and classic microbiology that provides us a crucial answer to a devastating ecological problem. And it just goes to show what might be old is again new. So this is the snippet for this week. I thought it was a good way to start off our semester of microbiology. And this takes us to our next paper.
Hold
that
on.
Mark,
Can I say that they also cite a paper from 2001, Lambert et al., that demonstrated that this particular species of Vibrio do make an extracellular cytotoxin, which draws a parallel then with Vibrio cholera. Probably many of our listeners have heard of cholera, a terrible watery diarrhea disease that can spread very quickly through populations, and it too is triggered by a cholera toxin. So
I wonder if it's phage dependent like the cholera toxin is, and the phage is regulated by the salinity and other environmental factors.
in other words, the Vibrio acquired the toxin through a phage. Yeah, horizontal transfer. Yep.
Yeah, so there's more to come, I'm sure, from this group. And this group is a powerhouse of marine microbiologists out of the Pacific Northwest.
So I had a question. I know a lot of people have been looking at this for the past 10 years, and there have been times where people lean toward a viral source for it and a polymicrobial source for it. And I wonder why these folks were able to find such success compared to other people.
I don't know the
answer. They
answer
do point
to
out
that.
in the discussion that it was necessary to come into the laboratory and have isolated a diseased starfish population and then also healthy populations, and then do careful time control experiments. So they say that it can take over so quickly that it would complicate identification of the culprit.
And it wipes them out so quickly too, Mark.
Yep.
Yeah, and then they dissolve. And it wasn't just that beautiful type of starfish that have had versions of this. And I think anytime that we change the environment, we change the selective conditions, if you think about it. And Michelle, that was such a good point, because if you didn't catch it quick, as the animal dies, you're going to have lots of saprophytic organisms feeding on it.
Exactly.
And so a lot more to sort through. Yep.
I like that. I and it's funny because I was really surprised they were able to isolate it relatively readily, and then it also matched what they got, from their read-throughs. That's nice.
So for our paper this week, Mark's going to take us through a paper that was in Cell entitled Uncovering Phenotypic Inheritance Signals from Single Cells Using Microcolony-seq. So Mark, take it away.
I am really happy that you're here because this was quite the paper.
Oh, yeah.
And the first thing that I want to say is that, we're so used to looking at things in terms of phenotypic stability. And in fact, I just gave my little mini TED talk on campus because, Michael, no one wants me to give a TED talk. So my students have to hear it. And I have one about the heresies of microbiology, and this is one of them. So we're reductionists. We really enjoy one genotype, one phenotype, but life doesn't care about our rules. And the small masters of our microbial world agree. So I have to tell you, this really struck me because in my own classes and my labs with students, I have them isolate bacteria from nature all the time. And every time we do this, we find unstable phenotypes. Many wild bacteria show small versus large colonies on plates, and this is also true for some pathogens. And I'd read about it for years. And as I was thinking about this kind of thing, I thought about invertible promoters, slip misspaced pairing, or transposable elements. But here's the thing, and this is why we talk about, you've probably seen the t-shirt, "Don't ask me, it's epigenetics." And and there's some truth to that, because just for listeners, epigenetic changes don't alter the DNA sequence, but they alter expression. This is often due to methylation of DNA sequences. Now, I've been interested in this subject literally my entire academic life, and it's much more common than what we teach.
Mark, you can probably appreciate that the term unstable phenotype, unstable has a negative connotation. Like we scientists like things to be stable, but in fact, it adds versatility within the population, which I'm sure you're going to emphasize in your talk.
Well, I hate to tell you that when I first started running into this stuff in 1988, I used to call it a metastable switch because I was fancy.
You were fancy.
I was fancy. Okay. And today's article is great. They create a whole technology. It's called Uncovering Phenotypic Inheritance from Single Cells with Microcolony-seq. And this is by Feigenbaum, Ram, Yedidi, Geffen, Katsowicz, Nagar, Aroiti, Ronin, Bar Meir, Rosenshine, and Balabam. And they're at the Hebrew University in Jerusalem. And it officially appeared in the journal Cell September 18th, which is today. It came out a little bit before that.
And I want to point out that their graphical abstract, if you're not familiar with what epigenetic means, they have a great cartoon on the first page of the article that you can probably see pretty easily. And then their in brief statement really helps you walk through that graphical abstract. So you'll be on the same page with us all.
They make it very accessible.
And it has to be. You know, I make jokes a lot with students about what's called the MPU, the minimal publishable unit.
Yeah.
And I think there are 3 MPUs in here. Okay. I really do. But I understand why they included it. So this is a hefty beast of an article. It's dense and full of detail with tiny print. But there's so much information here. And I learned so much going over to the point where I was drinking from the fire hose. So I am so delighted that I have 2 colleagues with experience in medical microbiology to help me through it. And I think that this is something that every microbiologist is going to love, and it's going to irritate the mucus out of students.
Oh, yeah.
And the reason why is because we want things to be consistent and predictable. As Michael said, the graphical abstract on the first page is remarkable. They have a graphical abstract, they have highlights, and they have an in-brief summary. Now, I've got to tell you, every single bit of this is good and worth your time. So I'm going to read the in brief to you because I think this gives you some idea. This is like the view from 30,000 feet. And for this kind of article, I often have students go to this graphical type of abstract or an editor's summary that you sometimes see. So in brief, Microcolony-seq systematically uncovers inherited phenotypic diversity directly from infected human samples, revealing, and boy, do I love this, long-term genetic and non-genetic memory from the host environment. These findings suggest that bacterial memory is widespread and may inform the development of strategies to target the multiple coexisting subpopulations within infections that can evade treatment. And I want to say the thing that got me going so much was resetting all of this, which we'll talk about in a minute. And oh, do I have questions for you two about this? And there may not be answers. But I've got to tell you, as I was going over my notes for it, they do a really nice job talking about phenotypic heterogeneity. And I'm really happy that Michelle, that unstable thing, because you're quite correct, that sounds negative, right? And it's history. So because some of the things that I look at in my little undergraduate lab involve heterogeneity and phenotype, You can imagine how excited I was by this. And every student thinks that a colony forms from a single cell and that colony is genetically homogeneous. It's not so. The outside of the colony is different than the inside in terms of selective pressures. And they actually, in the paper, the researchers, I shouldn't say they, the researchers give some examples why it's not a lab artifact that we're looking at. Because this kind of phenotypic heterogeneity, it's kind of a mouthful, but it's working. Transient resistance to antibiotics, differences in growth rates, surface structures that help with adhesins, all of these things which can relate to pathogenicity. I do want to say parenthetically that this isn't just about disease. This is about adaptations to changes in environments. We tend to take pathogenicity personally. And I don't think that's a bad thing at all, but this is not about causing disease per se. This is about organisms trying to gain the upper hand to produce more progeny. Because they knew there was such heterogeneity, at first they thought, well, we got to look at single cell RNA. And that's actually really hard to do with bacteria. And so what they decided to do is ask the question, could they investigate these stable subpopulations that arise during growth? Now, this type of heterogeneity can lead with division of labor, immune evasion. This concept I love called bet-hedging, which means that you might get a population that appears to be less fit, but if the environment changes, they're much more fit. And colonization of surfaces.
And we see that with the lottery, where the syndicates out of England effectively bought every number in the lottery so that they would win. And that's called bet hedging. And that's exactly what the microbes are doing here. Yeah. They're literally buying every number in the lottery.
Well, they did it first, right? I mean, that's the thing. So let's talk about EPEC a little bit. Enteropathogenic E. coli. And I believe, I want to get the name right. I believe the co-author with the last name of Ronan is the one who had found some of these things to begin with. That this is one of the situations I alluded to where you have large colonies and small colonies. And they have different amounts of disease that they're going to call. I mean, Arturo Casadevall doesn't like the word pathogen, but you and I can talk about pathogenicity and virulence, and that's fine. But what's interesting in this case, to get ahead, is that these changes, which are seen originally with small versus large colonies, get reset during stationary phase. And that's the part that blew my hat off. Because because we know a little bit of what happens in that transition to stationary phase, there are ways to look. So when they use the word, and they use it repeatedly, that bacteria have a memory and then they forget during stationary phase, they're talking about these epigenetic changes. Acinetobacter can do the same thing with opacity of colonies with different types of virulence. And they wanted to do this without visual cues. That was the important part to what they're doing. So you could do single cell RNA-seq. It's possible, but it's destructive and you can't preserve anything. So they wanted to create something they called Microcolony-seq. I guess people ran out of omics. Right? And as I'm reading this, I'm thinking, when do bacterial colonies enter stationary phase? And I realized I didn't know enough about that. It's an interesting thought. But what they're able to do is use Microcolony-seq. And if you look on, and figure 1 is a good example. They show you the workflow. And what they're looking at are microcolonies. Microcolonies. And what they can do is save part of that colony is frozen, and then they can analyze some of it. And that means they can do RNA-seq, and then they can also do whole genome sequencing. So this enables them to identify changes that take place genetically and epigenetically, while still maintaining a stock of the original subpopulation. And when I say microcolonies, they're 500 microns across. So I don't know how many cells that is. I want to say a couple thousand, but I'm not sure.
It depends what they're feeding them. It depends
Right.
what they're feeding them and how big they get per unit time, and it's many variables are determine how big they're going to get.
But
So
fortunately, the analytical methods are so sensitive, they didn't
Yes.
need a large population to do
no,
each of these.
and they actually, if you go back to the possible limitations of their work, speaking of that, by the way, Michelle, I'll come back to where we are, but I love how you said that. They said that they might miss tiny changes, and I laughed out loud because to me, they're looking at tiny changes, but they're even looking for even tinier changes in the future. And that's again because what you and I call a small population, that is to say a microcolony, something of that nature, is not homogeneous. And that's going to really cause a lot of students to throw their pencils in the air. I shouldn't
Well,
say that they're
or learn to
styluses.
really admire bacteria. They're very,
Yeah.
Well, I'm
very
there.
much more
I'm there.
sophisticated than you might think.
Absolutely. So figure one is amazing, and I know that, that I talked about it already a little bit, but there's a lot going on. There's a lot of statistical analysis, but it's one of those figures that the legend to the paper spans 2 pages. I mean, the legend to the figure, I should say, spans
Yes.
2 pages. And I can't fault them because they really did a good job explaining everything. So when I look at figure one, and I really wish people would start, part A actually shows again the workflow and how they're separating things. And then they actually go to B, which is all about their computational work. And I would draw your attention to section D of that second box, where they're able to show that there was heterogeneity in that microcolony with regard to genes that are turned up or down. Okay? And you go to C and you can see that you can really see the subpopulations there. Now, what are they looking at actually? Now, the EPEC strain that they're using forms 2 colony sizes, and I love how they describe them in all caps as small and big. Okay? And I
Technical
was
terms.
no, what else would you call it, right?
No, I love it. It's great. Make it accessible.
And what I thought was interesting, because I haven't thought about this in a long time because I have no real background in medical microbiology. So they grew it up and they said they're going to grow it in conditions that I'm not making fun. I'm just saying that it was very formal, that are relevant to the disease causing state, and they used DMEM media, and that's Dulbecco's modified eagle medium.
For tissue culture, which is people.
That I used to use when I took virology. Exactly, right?
It's people medium.
Well, it's not homogenized people, to go back to Elio Schechter.
No, it's not.
So they grew up the EPEC strain in DMEM, and I've got to say, this is one of those materials and methods sections that goes on and on and on. And I think that's because they want to make sure that if anyone wants to repeat it, they can. They went into a lot of detail here. So they grew it up in DMEM for 3 hours, and they knew already that that would induce virulence genes. Then they plate and they take those tiny microcolonies on plates. And it's important to note you couldn't tell big versus small at this stage. Again, they wanted something that was independent of the different morphotypes.
And they're also playing games with oxygen too, because they're effectively mimicking the environment that the microbe is going to find itself in. And the small intestine is not an aerobic environment like we incubate many of our plates.
Right.
It's an anaerobic environment. So they were very careful in describing the conditions that they're going to present the data for.
No, I think it's because we make assumptions all the time.
Mm-hmm.
And they were looking for what they're going to call virulent signatures. That's like in 1D. And one subgroup had some genes turned down, and the flagellar and chemotaxis genes are those. And other had genes turned up. And it's nice to know the type 3 secretory system, makes sense, is turned up. But what's cool, and I want people to understand, is because of the way that they did this, where they had some specimens still alive, as well as nucleic acids, they can actually look to see if there were changes at the DNA level or not detectable, meaning epigenetic. And I think that was remarkable for that. And again, they found that stationary phase appears to reset the system, which is amazing to me. They went on, the researchers, to see if they could detect those kinds of changes, this differentiation within microcolonies with other host-associated conditions. As Michael says, lack of oxygen, the addition of butyrate, or even stool components. And the less virulent subpopulation, which they call avir,
rightly so,
had greater motility and a higher salt tolerance. Now, when you hear that, you have to take a step back and think about selective agents, what's going on. And I think that there are levels after levels with this. And I don't know that I'm wise enough to draw the distinctions. But what I'm noticing is that even in this relatively modest number of cells compared to what I would see on a culture dish or in a tube, there's incredible plasticity. They have the ability to switch genes off and on without changing DNA sequence and to match certain microenvironments or other conditions that act on them. Now,
and if I could just point out
please.
Fitness advantages, they found the flagella and chemotaxis were turned down, whereas type 3 secretion system and other virulence factors were up. And it makes sense because to make either the beautiful flagella or the type 2 secretion system is expensive. They're multi-component protein-rich structures. So, of course, if you're in an environment where you're now about to attack the host and scavenge nutrients, you don't want to be also wasting your time or swimming away. So it's really beautiful that the flagella and chemotaxis are shut off in one population, and the other population, they see that the virulence factors are up.
And that same pattern occurred under different kinds of places where you would expect to see that happen. And that's always nice to see. And I don't want to speak for the authors, but I suspect that was a little bit why they went into the detail that they did, because I would be so suspicious of something that looks so beautiful. I mean, if you look at, and again, in figure 1 on F, they actually looked at some of these differences in adherence to HeLa cells.
Mm-hmm.
Yep. And they're very rigorous, very rigorous.
I wish there was a stronger word than rigorous.
Mm.
Because they were, and it reminds me of someone who's trying to prove something that they don't think is going to be believed. And I'm not making light. I'm saying they really did an incredibly exhaustive job, it looks like to me.
I agree.
Now, I want to say that they did see some phase variation, that is to say, invertible DNA segments, which they were able to pick up when looking at DNA sequences, but a lot of these epigenetic changes as well. And they don't really know initially where those things are taking place.
And let me just elaborate. An example of
Please.
an epigenetic change would be, for example, you can methylate particular residues on the histones, the chromatin. So you basically can have the DNA tightly packed so it's not accessible to the transcription machinery, or more loosened up. So those are changes that don't require you change the nucleotide sequence, but you rather change how the DNA is packed to turn it on or off.
It happens in bacteria as well as in you and me. And it's interesting to note that how long those epigenetic changes last is a really critical thought. And I always wondered in my heart of hearts what controls that. Because it isn't just like one round of replication, because you would think you would get rid of things in like 1 or 2 rounds of replication. It's not. And that's what's so interesting to me about it. There have to be some things controlling it, which they actually had some success, I think, finding some useful things to look at. Now, again, the reason they looked at this particular system is they knew a little bit about this EPEC, and I feel like shouting when it's uppercase, big and small, right? But they were able to find out a lot of things as we've just been talking about here. I don't know as much as I might about the issues involving their other 2 systems. So I'm asking for people to kind of help me out with that a little bit there. Oh yeah, oh man, they've got so much stuff going on here.
But we don't have a lot of time.
Yeah. No. So what I want to say, and thank you for that, because this is always worrying me, because I was so enthusiastic and I learned so much. I want to say that the lessons with the EPEC strain are the important ones. There's something about stationary phase that resets it. And I thought this was especially interesting, Michael, because I got really curious because they kept saying that the PER, A and B genes were involved in that resetting, and they have a wonderful experiment to try and show that, to keep it from happening. And I noticed, okay, PER A is a transcriptional regulator. All right, I'm with that. And then, like PER B, we don't know what it does from where I can see, anyway, in E. coli. And that tells me that they might be on the track of something that's controlling that resetting of the epigenetic system. Because again, if you're just looking at methylation, that has to be changed at the next round of replication and at the one after that. So that change that you made that affected transcription would go away. And so there's something maintaining it. And I look so forward to that. Years ago, when they first started looking about methylation and pathogenicity, Bruce Lowe did that stuff, and it just amazed me. So they also looked at a medical isolate, a UTI, urogenital tract E. coli infection. They did that kind of work from urine, and a really sad Staph aureus example causing bacteremia. Now, both of these are clinical isolates. They haven't been, as I talked about today in class, domesticated, because this EPEC strain has been used for some time. What's interesting about this to me is that they found many of the same ideas. That is to say, that what you think is a small population that's uniform isn't. There are subpopulations within it, and some of those subpopulations can benefit from a particular environmental condition. And this gives them great variability in dealing with a variable environment because, as I told students today, we are a great series of ecological niches walking around.
And the UTI experiment Mark just is alluding to is in figure 5. And again, what these authors are great at doing is walking the reader through their thought processes, through their experimental setup, so that you can stay with them. I'm reminded of our time at the undergraduate conference last year when we heard about how students have to effectively go off and summarize the paper after we discuss it. And I believe this cell paper happens to be open access. And so you'll all be able to follow along and look at this in the greater detail and really discover each of these figures, because each figure, as Mark said, is a paper unto itself, because you can really dig your teeth into it and really understand what the principal component analysis is looking at and what you're looking for UTI subpopulations and how the antibiotics are actually resulting in the differential of the microcolony appearance.
Yeah, so 3 different biological systems are microbiological systems, but all pointing to the same theme, that there is this subpopulations within the larger population, because bacteria, of course, just want to maintain the lineage.
Yes.
Not like humans. It's the individual human.
There's a very old piece of work, Michelle, and this is discovered many, many years ago by Bob Bielis and Linda McCarter. There is a bacterium. It's now called Pseudoultramonas atlanticum, and it has 2 forms. One, it makes copious exopolysaccharide. The other one, it does not, so dry and wet. And the sticky form is very adherent. And, every 1,000 generations or so, the division will produce something that's dry, and then that allows them to move to a new location in the ocean. And then as it's planktonically replicating, every 1,000 generations or so, you'll get a sticky one. So that's how it's dispersing. And that's due to a transposable element that jumps into and out of an EPS gene precisely, which is relatively rare for transposable elements. And, that always got me thinking about variation. And so
EPS is a polysaccharide-like coating.
Right.
So again, it's a population effect.
When you think it's not, but it's not.
and I loved, in getting back to this paper, the Staph aureus example with iron limitation, because that's something that people tend to forget. that most of our iron is tied up in transferrin and lactoferrin, and that's a good way to keep potential disease-causing organisms at bay. But that's an opportunity. And maybe that's one of the take-home lessons here. It's an opportunity. So what we think of in a culture, just as the microbial universe is vast and various, so is a colony. And even a small colony. And it makes me really sit and think, what am I measuring? And it reminds me of what Bill Metcalf at Urbana, Illinois used to tell me, is that a phenotypic assay is for your benefit, not the bacterium's.
That's correct.
And he's right. So I wanted to summarize what was really important here, because I think that there's so much detail, so much stuff to chew on, but these 5 take-home messages are really, really important. That they're apparently shifting in multiple states within a population. And this Microcolony-seq allowed them to find that out. And I'm very suspicious of things that form colonies, because I think that's a form of selection. But in the last 2 examples, they weren't growing them a lot. They were taking them from human patients and then putting them on the plate. So they weren't passaging colony to colony. This epigenetic memory of host-related condition occurs within subpopulations. So there's variation taking place. And I don't know how extensive it is. I do know that this approach could be used with any type of system. My own with bioluminescence, this would be a really good system to do if I had that kind of setup. Go ahead. I'm sorry.
Their last paragraph of their manuscript, implications and future prospects. And here I'm quoting them: by providing a global landscape of genetic and phenotypic heterogeneity and its phenotypic consequences.
Yeah.
This Microcolony-seq method allows for the systematic identification and characterization of inherited phenotypes without the need, underscore that, without the need for genetic manipulations.
Yep.
And so they then drop the bombshell on us. Our method should pave the way for detecting many more differentiation strategies, hence Mark's concept of bioluminescence, used by colony-forming organisms, including fungal pathogens.
Yes.
So it really is a remarkable piece of work that this group has produced. It's extremely provocative and really drops the gauntlet down as we begin a new semester of learning.
It's also exciting to appreciate that diversity within a population actually makes the population overall stronger and more fit, rather than homogeneity.
Now, I like what you're saying, and I know what you're saying beyond there, but just as before, and it's important to note, it doesn't have to be about something that causes a disease. That's the thing, right?
Oh, absolutely.
Right.
Any type of phenotype, I make a joke that the hand of Darwin is on all that lives, and this is a great example. So as you just heard, this idea of multiple states, this epigenetic memory, the resetting of memory in stationary phase, I really want to emphasize that. I want to know more about that.
About the why or the how?
Yes.
If you think about it, stationary phase is really a population effect where death equals life. But you're asking for what is stationary phase mean from an individual. And that's where the epigenetics comes into play. And we haven't had any deep think on that until papers like this are emerging.
So,
but also if you think about log phase versus stationary phase, basically bacteria will keep growing as long as they sense the conditions are favorable, that one bug can make 2 and 2 can make 4. When conditions are no longer, whether they run out of food or some environmental
waste,
then they not going to make another cell. I'm going to go into a more hardy, resilient cell type and just wait it out.
And I was really influenced by growth advantage and stationary phase work, by Coulter originally, and then Finkel. Fascinating stuff that suggests that, quote, stationary phase is far from stationary. There are constant waves of mutation as individual. Is this sounding familiar?
Yep.
Individual cells begin to have advantages within that context. And this is like not about disease. That's my big fear with this kind of paper, is people think it only applies to pathogenicity. And that's not the case. It applies to everything microbial. And again, this heterogeneity in this paper is about avoiding the immune system and dealing with stress. Yes, but it doesn't have to be about that. And but getting back again to our own wish to improve human health, the fitness advantages of the subpopulations that they find could help develop therapies. And I love the fact they even stated their limitations of their work so clearly.
Oh, yeah. And for many of our students out there who are beginning their experimentation into the production of beer, this has great implications in the fermentation world.
Yeah.
Because you start out with an inoculum that hopefully will give you the same flavor profile, the same off tones that you want, the same aftertaste that you're looking for, the same concentration of alcohol, all of which can be subject to these microcolony variants. And you have to make certain you got everything tuned right. And so beer making is really even more remarkable when you think about it, that you get the same type of product each and every time.
I was so amazed by this paper. And Michelle, I want to say thank you for kind of limiting my enthusiasm for the other two systems, because I think
not your enthusiasm, but our ability to share that enthusiasm.
No, I think it's important, because the thing is, I think if you understand the first system, the EPEC system, if you look at that and it's so darn clear cut, then you can start to apply that to other stuff like we've been doing.
Yeah.
So Michelle, I wish you could adopt me.
Uh-oh.
Yeah, and I wish I had more time today.
Yeah.
Well,
but I don't.
Thank you both, Mark and Michelle. Our show notes are at microbe.tv/TWiM. If you like our work, consider supporting us financially. You can go to microbe.tv/contribute. We'd love to get your questions and comments, and you can direct them to twim@microbe.tv. And Michelle Swanson is at the University of Michigan. Thank you, Michelle.
Thank you. It's always a pleasure.
Mark Martin is at the University of Puget Sound. And thank you for being adjacent today, Mark. We really enjoyed your contribution and the selection of this fascinating paper. So thank you, Mark.
You're welcome. My mind was microbially blown.
And I am Michael Schmidt, and I'm at the Medical University of South Carolina. Vincent and Petra will hopefully be back with us from their travels on our next episode. And I'd like to thank ASM for their support of TWiM, Ronald Jenkins for the music, and this episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. See you next time on This Week in Microbiology.